Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor. Welcome everybody again to another episode of our show. Today bring you another really awesome guest involved in creating a better tomorrow. Today we have the honor of being joined by Dr. Jonas Hannestad, who is Chief Medical Officer of Gain Therapeutics. This is a really interesting company that's focused on the discovery and the development of various groundbreaking medicines that ultimately can transform lives, provide hope, improve quality of life, specifically to people suffering from really debilitating neurodegenerative diseases. And they're also focused on rare genetic disorders and cancer. Dr. Hannestad has been with the company since just a couple months ago. He brings 25 years of translational and early clinical development experience in the central nervous system space. His academic work focused broadly on immuno-neurology, particularly on the effect of systemic inflammation on myeloid cells in the brain, and his drug development expertise spans multiple indications, including Parkinson's disease, Alzheimer's, and ALS. Previous to this role, Dr. Hannestad served as Chief Medical Officer at both Tranquis Therapeutics and Capacity Bio, as Senior Vice President of Clinical Development at Alkahest, as Medical Director at Denali Therapeutics, and as Director of Neuroscience Discovery Medicine at UCB Pharma. Prior to his industrial career, Dr. Hannestad led Clinical Neuroscience Research at Yale School of Medicine. He did his postdoc work and residency at the Bristol-Myers Squibb, Duke School of Medicine, and UCLA Neuropsychiatric Institute. And then he did his Ph.D. in cell biology from University of Messina in Italy, and his medical degree from the University of Oviedo, Spain. A lot of really interesting topics to get into today. Honored to have him with us. Dr. Jonas Hannestad, thank you so much for taking the time to come on the show.
Thank you. I'm very excited to be here.
I'm excited to have you. A lot of really important themes today. And, as we typically do, I love to give the guests on our show the floor to begin with, to talk a little bit more about their background. In your case, one of the fascinating areas, obviously you studied medicine, you studied psychiatry, but then sort of very early on in your research, this really interesting intersection of neuroscience and immunobiology comes into play, studying these concepts that are quite popular nowadays, actually, in terms of the inflammatory system and its effects on our brain. Talk a little bit about the early days, if you would, and a little bit about how you got into that early part of the research program, if you would.
Yeah, certainly. So very early in my career, I realized I was interested in research, and I would say, this probably happened even before I started medical school. But while I was in medical school, I saw that many diseases did not have good treatments, and I realized that the best way to improve that was to do research. And since that was something that I was interested in, found fascinating, I wanted to dedicate my career. So I decided already in medical school that I wanted to do mostly research. Now, what exactly I wanted to do, I didn't know at the time. I was, fairly naive at that stage in my career and that age. But I started working with one of my professors at the medical school, and this was in Spain. Mm-hmm. And he was an anatomy and histology professor. And we worked on this project where we looked at expression in immune cells of certain proteins that were typically believed to be present in neurons. Yep. And that project, which eventually led to my Ph.D. in cell biology, that was what opened the door to this whole field, which was a burgeoning field at the time of neuroimmunology or immuno-neurology, however you want to call it. But the idea that the brain and the nervous system is not isolated from the immune system and from the rest of the body, but they're very closely coordinated.
Mm-hmm.
So that has been I guess a sort of a thread of my career all along, with certain periods of a little quiescent perhaps, but it's something that I've either worked on or been following very closely.
Mm-hmm. And alongside of that, another really interesting piece in your portfolio, as you know, I spend a little time sort of swimming through your publications, is this whole application of imaging technologies, sort of the cutting edge PET and so forth, in specifically the field of broadly psychoneuroimmunology. You have this really cool paper on the application of PET imaging in the field, and then several papers on everything from studying single cell transcription changes and synapses, to sort of looking at tau in Alzheimer's disease, and then sort of broadly at brain pathophysiology. Talk a little bit about the role of imaging and some of these imaging technologies along the way as you were doing your work in this space, because clearly they sort of are companions in many ways, and I'd love to hear that part of the story as well.
Yeah. So the early in my career, so after my PhD, when I did a postdoc at UCLA, and I decided that this neuroimmune interaction was something I really wanted to work on. At that stage, I was working on mouse models, mouse and rat models for the most part, and there you could do experiments where you sacrifice the animals at the end of the experiment, and you look at their brains with various methods, right? But then when I transitioned to doing more clinical research, and I realized that obviously that's very challenging in humans for obvious reasons. So I uh, became interested in neuroimaging as a way to understand what's happening in the brain while at the same time manipulating the immune system. So the paradigm that I developed, and this was when I was a faculty member at Yale, was to use injection of a small amount of what's called endotoxin or
Mm-hmm.
lipopolysaccharide, which is basically part of the outer wall of certain bacteria. When you inject small amounts of that into the bloodstream in humans or in animals, the immune system reacts as if there were an infection with that bacteria. So there's a huge reaction, and that reaction not only affects bodily functions to some extent, but also that it has effects on the brain. And this was well known based on many, many years of research predating me, obviously. And part of the research that I did at UCLA was using this paradigm. But then what I did as when I was at Yale was to combine this paradigm in humans with brain imaging. So specifically, I used a modality called positron emission tomography, which is
Yep.
it's a way to image the brain, to image specific molecules or targets in the brain. So you have a pharmaceutical molecule that's labeled with a radioactive isotope,
Mm-hmm.
and then when that binds to certain targets in the brain, it the image you get can sort of it lights up, if you will, certain areas, and then you can use that to quantify how much or how little there is of that specific target. So what we did was we recruited people to receive LPS injections or endotoxin injections and have this acute sickness phenomenon occur in their bodies, and it's pretty short lived. And then we would use brain imaging, PET imaging, to look at things like just overall neuronal activity, see which parts of the brain become activated by this, which parts become perhaps deactivated. And then we looked at a cell type called microglia, which are immune cells that reside in the brain and that are there for reasons of fighting infections or repairing injury. And they clearly became activated when the immune system in the body thought that there was an infection, perhaps in sort of preparing for that infection to entering the brain.
Yep.
And then the last thing we looked at with this endotoxin paradigm was dopamine levels. So when you probably experience this if you get the flu or you get COVID. One of the prominent symptoms is fatigue, and this lack of interest in doing things and just bodily fatigue. And our hypothesis was that this fatigue probably was related to dopamine in some way. So we used PET imaging to measure dopamine changes in the brain during when people were experiencing sickness behavior from the endotoxin injection.
Mm-hmm. Mm-hmm. Excellent. Well, yeah, it's again, I wanted to highlight those two things because obviously they're very, both of them in terms of the immunobiology of the CNS disorders, and then of course where we're headed with all this complex imaging technology, imaging individual cells and transcriptomes and all that. So I wanted to lay those two things out there just to give a little bit more about your scientific background. And then from there, and we'll get into GAIN in a minute, but from your research, you get into the biotech and pharmaceutical industry. And in the time that you spent there, prior to GAIN, you developed quite, a significant experience in all sorts of therapeutic interventions. So obviously you worked on a lot of small molecules, medical devices. I saw you involved in a nicotine vaccine. You're working with these really complex plasma fractions at Alkahest, that's a whole other complexity that we don't normally get into in drug development or traditional drug development, at least. Talk a little bit about some of your experiences here and a little bit of how you saw sort of the pharmacotherapeutic themes, let's say, as it pertained to the CNS develop over your time in this space, because clearly CNS is a unique area. We got a blood-brain barrier. We have all sorts of other unique aspects of it in terms of pharmacotherapeutic interventions. Talk a little bit about your past history in developing some of these therapeutics, if you would.
Yeah, so maybe as a segue, I can just for the audience to explain how I got from clinical neuroimaging research in academia to drug development. And the reason for that is that neuroimaging is used in drug development in several ways, right? So one obvious use in CNS drug development is to use imaging for diagnosis to make sure that you have patients with the right disease that you want to test a new treatment for. But then more interestingly, what I did early on in my industry career was to use PET imaging specifically to measure the ability of certain experimental drugs to get into the brain and engage with the target. So what we call target engagement. And that's something that PET is very helpful for. And then another component of imaging in drug development is using imaging endpoints. So measuring some biological process in the brain with imaging that we believe is relevant for the disease, that may be more predictive of progression or improvement than measuring what we call the clinical endpoint, right? The eventual symptom or sign that the patient experiences. So that's how I sort of got into drug development. But then, yeah, as you mentioned, within drug development, I've been working on many programs across both mid-size pharma and small biotech, and the programs have spanned a wide array of diseases. So mostly focused on neurodegeneration. So that I usually say that the diseases that I know a lot about are Parkinson's, Alzheimer's, and ALS. Mm-hmm. But I worked on other neurological diseases, for instance, epilepsy. I did a lot of work on that when I was at UCB. I've worked on non-CNS diseases, including ophthalmology, even dermatology and kidney disease, various things. And then also, in addition to these disease areas, I've worked on different modalities, which you alluded to as well. So mostly small molecules. So small molecules are things that you can typically take, in tablet or capsule form that are orally bioavailable. Also worked on biologics, so antibodies, and then the plasma fractions that you also mentioned at Alkahest, which is a whole story on its own, that maybe for another time, and a device that we were trying to develop for people with on dialysis, so people with kidney disease on dialysis who had cognitive impairment, and there's a specific protein that the dialyzer membrane was not able to remove that we believed was involved in cognitive decline, which occurs commonly in these patients. And then with this device that we had developed, we could remove that and perhaps improve cognition.
Excellent. Excellent. Awesome. Well, now with laying, that framework, let's move on to Gain Therapeutics because it is a really interesting company located down the road here in Bethesda, Maryland. Here you're back sort of in the small molecule space, developing interesting substances that cross the blood-brain barrier. And in this case, a very interesting target in terms of this glucocerebrosidase, which is something we normally, this lysosomal protein that we hear a lot about, we heard a lot about over the years in terms of Gaucher's disease, and there's sort of the area of liposomal storage genetic disorders. Give us a little bit about sort of the background story here, and a little bit about how the company made the initial connection that, hey, this is this target, which has been studied for a while for all these other interesting genetic CNS disorders, may have an application in Parkinson's pathogenesis.
Yeah. So just for the audience, the background now. So GBA1 gene is the one that's if it's mutated in both alleles, and you have basically a lack of the enzyme of glucocerebrosidase, then you develop Gaucher disease. But what was discovered about a decade or so ago was that if you have one mutation in this gene, then you have enough enzymatic function that you don't develop Gaucher disease, but you have an increased risk of Parkinson's. And it's actually the genetic risk factor that's the most prevalent most common for Parkinson's disease. So this genetic connection, that you have a mutation in a gene that obviously predates anything else. So there's a causality there, not just a correlation. This genetic risk leads to an increased risk of Parkinson's. Then there's something about this mechanism, the GCase mechanism, that makes those specific cells in the brain degenerate more readily. And they're, the story is quite complex. So GCase metabolizes certain lipids in lysosomes, and those are the ones that accumulate in Gaucher disease when you don't have enough enzymatic function. In Parkinson's, it's a little bit more complicated because you don't typically accumulate a lot of those lipids, but you have other effects of these mutated enzymes, including you have effects on the endoplasmic reticulum, which is kind of the transport system for proteins in the cell, if you will. You have stress in the lysosomes, where GCase typically resides, and you even have stress in mitochondria, and those are all cellular organelles or cell systems that have been implicated very strongly in Parkinson's. So mitochondrial dysfunction and lysosomal dysfunction appear to play a role in Parkinson's pathophysiology. So the fact that those are affected by a GBA mutation is how we can connect those two. And then the other part I'd like to add is there's also a connection between GCase and alpha-synuclein. So when you have a mutated GBA allele and a mutated form of, part of your GCase is a mutated form, you have abnormalities in alpha-synuclein. And this could be the explanation for why this is linked to Parkinson's, because as you know, in Parkinson's disease, accumulation of misfolded and aggregated alpha-synuclein is the hallmark, the pathological hallmark when you look at brain slices, but it's also probably very heavily involved in the pathophysiology and the causing of the disease. Okay. The Gain's lead molecule, GTO2287, is a molecule that we have found it binds to GCase, and it helps this misfolded GCase go along into the lysosomes and and thus reduce the stress on both the endoplasmic reticulum
Mm-hmm.
and the lysosomes. And that has a range of beneficial effects that we have detected in both in cell systems and in vivo in animal models. So
Mm-hmm.
it reduces, as I said, mitochondrial dysfunction, lysosomal stress, but also inflammation, which is also seen in Parkinson's disease. It reduces the accumulation of alpha-synuclein. It increases the survival of these neurons that are typically affected in Parkinson's disease, and it even reduces something called neurofilament, which is a biomarker of neuronal health that is commonly used in drug development.
Mm-hmm. Mm-hmm. And I appreciate you walking us through the biology there. And so in getting to this point where you realized, hey, we have this G202287, it can restore the function of this protein. And along the way, to getting there, some really interesting computational biology came into play here. And we've been profiling AI and some of these tools for a while now in drug discovery, but in this particular case, working with the Swiss National Supercomputing Center, and I think some NVIDIA tools were involved, quantum simulations, all this. Talk about this, because I love hearing about interesting candidates that pop out of these tools, not just the tools themselves. So walk us through a little bit about how GT02287 ultimately came to fruition via some of these computational tools.
Yeah, so this is basically how the company was started. So it was this computational model, which again, I'm not my area of expertise. I'm probably going to
Cool.
butcher this, but I'll tell you what I know. But it's basically a way to use computers and very complicated software to model the interaction between proteins and small molecules. And this is something that traditionally, say, 20 years ago, they had to be done in the wet lab and with
Right.
actual experiments. But with the advent and the progress of computing power, we can do a lot of this now in these computer models instead of having to do the actual experiment. And basically, the way it works is that it would look at the protein that's of interest. In this case, it was GCase, but Gain has applied this to many other proteins as well in
Mm-hmm.
our preclinical pipeline, and in theory, you can apply it to any protein for which you know a lot about the structure and the amino acid sequence and those things. And then the computer will basically test hundreds of thousands, if not millions, of compounds, and these could be real compounds that exist that have been made, but it has the information about them, and test whether they would bind to any part of the protein. And you can even in the sort of the second iteration of this model, now we can test a larger chemical space, if you will. We can test theoretical molecules. So a molecule that may never have been synthesized, will this molecule bind to a certain part of the protein? And then based on that information, you can then identify molecules that either real molecules or theoretical ones that
Mm-hmm.
bind to the part of the protein that you want them to bind to and have the intended effect, at least in silico. And then the next step would then be to test this in reality and see you make the molecule, you have the protein, you test it in the lab if the prediction is actually real. And that's how this lead molecule was discovered in essence.
So again, complex computational biology tools to discover the molecule. You've been doing an exceptional array of in vivo, a lot of animal work on neuroprotection, on the restoration of motor function, and all of these interesting preclinical models. You recently went into a phase 1 clinical trial, and I think that's still ongoing. That we're not going to know much about that until Q3. But can you just tell us a little bit about um, what's happening now, sort of on the development front per the compound, and where we are in as we enter May?
Yes. So as I said before, the compound was developed using this computational platform, and then it was obviously characterized extensively, right, in both in vitro and in vivo, as we spoke about before. Not just to understand how it interacts with the target, the GCase, and these other cell systems, but also tested for safety, right, before you take it into humans. And then, so at the around sometime last fall, the phase 1 study started. So this and this is public information. So this is a fairly typical phase 1 study in healthy volunteers of men and women who have no known medical problem, and they will take either a single dose of the drug at escalating doses, and that's the single ascending dose phase, or multiple doses of the drug, so for 14 days. And the main purpose of the phase 1 study, just for the audience, is to test the safety and tolerability of a drug for the first time in humans, to make sure that the doses that you intend to give are fairly well tolerated and that they're safe, and then characterize the pharmacokinetics, which is basically the way the drug behaves once it gets into the bloodstream. So you want to know about drugs that are given orally, you want to know that enough of the drug gets from the gastrointestinal tract into the blood so that it can distribute it to wherever it needs to go, in this case, the brain. Because knowing how much gets into the blood, you can also use that information to estimate how much gets into the brain and whether you have enough drug in the target organ to have these effects that you have observed in animals. So right now we recently completed the single ascending dose part of the phase 1 study, and we dosed up to the highest dose that we intended to go to, which is at the upper end of what we believe is the therapeutic range, and that was well tolerated. And now we're in the middle of the multiple ascending dose study, which will complete sometime later, probably this summer. And then once we have the phase 1 data, so the safety and tolerability, we know how high we can dose, whether there are any specific safety signals that we need to monitor for, and then the PK and understanding how many milligrams do you need to take orally to get a certain amount in your blood. Then we would proceed into a patient study, and that's currently in the planning stages. And there's limited amount of information I can disclose, but in essence, it's a study in people with Parkinson's disease who take the drug for a period of 3 months, and then we would focus on measuring various biomarkers. So this is not a study where we look at slowing of progression because it's too short, and it's also not a symptomatic trial. So we don't believe that this drug has any symptomatic effects in Parkinson's. So all the drugs that are used for Parkinson's, they are symptomatic treatments. We believe that this drug eventually will slow disease progression, but as a first step in evaluating that, what we're doing is we're testing the effect of this drug on biomarkers. So the same biomarkers that we tested in animals related to GCase activity and levels, lysosomal function, mitochondrial function, endoplasmic reticulum stress, and inflammation, etc. We'll measure all of that to get confidence that our drug is doing in humans what we've seen it do in both cell systems and in animal models.
Excellent. And obviously, and we're obviously going to put the link to Gain Therapeutics in the bio of the show, but clearly the Parkinson's disease target is of course number one on the pipeline, and then you have a wide range of CNS conditions on here. Obviously, you have dementia, Lewy body dementia, Alzheimer's disease, and then you have a series of rarer genetic disorders. So we spoke about Gaucher's, which is on here. A couple other ones that I had to look up in terms of Krabbe disease and GM1 gangliosidosis, and here we're dealing with other interesting enzymes that are mutated in terms of galactosylceramidase and beta galactosidase 1. Obviously, this I mean, this fits into your specialty here in terms of both CNS disorders and the ability to get these novel modulators into the central nervous system. Can you just say a little bit more about the portfolio and some of your plans here?
Yeah, so the I think that the uh, the initial objective of the company when it was started was to apply this computational platform to understand interactions between proteins and small molecules, but with a specific focus on enzymes. And a lot of those rare diseases that you just mentioned, these are all lysosomal storage disorders,
Yep.
for the most part,
Yep.
where you have a deficit in enzyme. So in Gaucher, it's GCase, but in these other diseases, it's another enzyme that's mutated in some way, and it doesn't work properly, and then you have accumulation of something, some kind of substrate in lysosomes, and you have different sort of um, symptoms and signs related to that accumulation in each of these diseases. But the concept is that we use the computational platform to develop small molecules that can modulate these enzymes in a way that we believe is beneficial for these diseases. So for the lead molecule specifically, our intent is to go into Parkinson's, but also Gaucher disease, because the hypothesis is similar, right? It will modulate both wild type, if you will, GCase, and also mutated GCase. So we believe that GT-02287 could have benefits in Gaucher disease. So that may be a parallel clinical development path to Parkinson's. And then in addition to that, you mentioned dementia with Lewy bodies, and again, for the sake of the audience, dementia with Lewy bodies and Parkinson's disease are both diseases that are caused presumably by the accumulation of alpha-synuclein in the brain in different neurons. And the main difference, as traditionally defined at least, is that Parkinson's disease, you start with motor symptoms because you have alpha-synuclein in the substantia nigra and the striatum, which affects your ability to move smoothly and so forth. Whereas with dementia with Lewy bodies, you have this alpha-synuclein in more cortical areas, which will affect your cognition. You'll have problems with memory. You also have problems with hallucinations and so forth. But the underlying connection to GBA1 exists in both. Actually, the risk of dementia with Lewy bodies is actually higher even when you have a GBA mutation than Parkinson's. So there's a very clear link there, and therefore a third development path that we're considering is in people with dementia with Lewy bodies for the lead molecule. Now, the other molecules or the other disease areas that you mentioned, those are all still in preclinical development. So those are molecules that have been, so for instance, Krabbe disease, they have been made, and we're characterizing them in vitro and in vivo to make sure that they work on the enzyme in the way we intended, and then those will then potentially move into clinical development sometime in the future.
And just one other thing I wanted to ask you about, and if this is confidential, we don't have to talk about it, but I noticed down on the list here is a category of solid tumors, and it says it's sort of to be announced. But clearly, when we talked about lysosomes and lysosome storage disorders on the show in the past, a lot of that ultimately has connection to cancer and both development and progression. And we have, whether it's, the ability of lysosomes to get rid of nasty proteins that are folded wrong or initiate apoptosis and all these other important mechanisms. When they get all gunked up, problems occur. Anything you could say about cancer, or if it's just too early to talk about it, that's fine too. But with your immunobiology connection as well, I was like, wow, this is kind of coming full circle in a way.
Yeah. I think it may be a bit premature to talk about the cancer programs because, as you said, they are very early, and but that is definitely a focus of Gains, and it's more about the current strategy that right now a lot of the focus is on Parkinson's and the lead molecule, and the oncology focus is less of a priority at this time, but it's something that we have a lot of expertise in, and we have, as just as an example, one of the co-founders of the company is someone with a background in oncology, and one of the people in our scientific advisory board is the former head of National Cancer Institute. So we have it's definitely an important area of focus, but I think maybe premature for today's conversation.
No worries. We'll do it. We'll do it a follow-up episode next year on that one. But no,
Okay.
that's cool. Um, Jonas, while we have you, anything else coming up that we should know about? I know you've been pretty active doing some collaborations. You're working with the Michael J. Fox Foundation and some other Parkinson's focus groups. Any other things coming up in terms of milestones that are public facing, other initiatives for you in terms of conferences, talks, places that we may run into you and meet you and listen to you talk about the company? Anything else that I'm missed? Please take the floor.
Yeah, so I think you know well, this is so not as a representative of Gain Therapeutics, but it's sort of in my personal capacity. So I work closely with the Michael J. Fox Foundation, and we have this initiative called From uh, Targets to Therapies, and we have a meeting in New York City next Tuesday of next week, where we a series of experts in the field get together to help the Michael J. Fox Foundation prioritize a long list of potential targets that they have in Parkinson's disease, to narrow that list down so that they can get some targets that they can work on and validate as potential targets in Parkinson's disease. So I think this is it's something I'm doing, not related to Gain, as I said, but it's something that's very exciting to me because, as
Absolutely.
you know, Parkinson's is a disease that affects close to 1 million people now in the U.S. alone, right? And 10 million or so worldwide. And the only treatments we have are symptomatic. Now, symptomatic treatments are really good. They work very well, especially early in disease. But as the disease progresses, which it always does, these symptomatic treatments work less well, and they have more side effects, and people develop additional symptoms and so forth. So the ability of the field, pharmaceutical companies and biotech companies, but also organizations like the Michael J. Fox Foundation, to identify new targets that could lead to treatments that slow the progression of this disease is very important. So
Yep.
this goes beyond Gain's specific molecule, which obviously I have hopes that will do that, but you have to cast a broader net to
Yeah,
increase your chance of success.
sure. No. Such is the nature of drug development. But no, it's an awesome set of programs. Again, I wish you and the company the best with it as you continue down this clinical path, and obviously getting the some of the other targets, moving as well. No, really great story, and again, I'll look forward to doing a follow-up with you, maybe in a year or so time as the story progresses. But really, really great stuff. Again, for everybody that's going to be watching this particular episode on our YouTube channel or listening to it across the various podcast networks. Again, you've been spending time with Dr. Jonas Hannestad, Chief Medical Officer, Gain Therapeutics, developing these novel, groundbreaking medicines to help improve quality of life and transform lives of people suffering from these debilitating neurodegenerative diseases, as well as equally debilitating genetic disorders and potentially cancer. Jonas, I really want to thank you for taking the time to come on the show to introduce us to everything that's been going on in your career as well as at Gain now. Obviously, thank you for what you're doing and moving these products forward. And as we like to say on our show, thanks for helping to create a better tomorrow for so many patients out there via what you're working on. Really great story, and I definitely look forward to doing a follow-up with you.
Thank you. Yeah, it's been a pleasure, and I would be happy to participate in a follow-up interview in a year or so, and hopefully with some exciting news on our progress.
All right, be well.